Hello all, in this video we'll continue with now physiology. Now we'll see about receptors. Receptors are capsulated structures which contain sensory nerve endings. They respond to stimulus. This is also known as biological transducers, which convert stimulus into action potential. Classification of receptors. There are two types, extroreceptors and introreceptors. Extroreceptors are further classified into cutaneous receptors, chemoreceptors and telereceptors. Cutaneous receptors. The touch sensation is carried by the markle disc and the mucinous corpuscle. Pressure sensation is sensed by Pacinian corpuscle. Cold temperature by cross-end bulb. Warm temperature by Ruffini end organ. And the pain sensation is carried by the nociceptor or the free nerve endings. The chemoreceptors. For the taste buds are the chemoreceptor, and for the smell is sensed by the olfactory receptors. Now telereceptors. For vision, the receptors are rods and cones in the retina, and for hearing, the organ of cottae is the receptor. In extroreceptors, the stimulus is from the outside of the body, and in introreceptors, the stimulus is from within the body. Now we'll see about introreceptors. They are classified into visceroreceptors and proprioceptors. In visceroreceptors, the stimulus arises from the visceral organs, and in proprioceptors, the stimulus is the change in position of the body. Examples of visceroreceptors: stretched receptors in the heart, and baroreceptors in the vessels, chemoreceptors in the GI tract, and osmoreceptors in the urinary tract and brain. Now the proprioceptors examples are muscle spindle in muscle, and Golgi tendon in the tendon and ligament, Pacinian corpuscle in fascia, hair cells in the vestibular apparatus. Now we'll see properties of receptors. First is specificity of response. It is called as Muller's law. It refers to the specific type of receptor as first specific sensation. Example, the pain receptor only senses the pain sensation. Now, the sensory adaptation or desensitization is another property. On continuous stimulation of the receptor, there is decline in discharge of sensory impulses. There are two types of receptors, the phasic receptors and the tonic receptors. In the phasic receptors, they get adapted rapidly, and in tonic receptors, the adaptation takes time. Next is Weber-Fechner law. The response is equal to constant into logarithmic of stimulus. It states that the intensity of response is directly proportional to the logarithmic increase in the intensity of stimulus. Now sensory transduction. It is the conversion of a stimulus into an action potential after sensing by the receptor. Now receptor potential. It is non-propagated and it does not obey all or none law. It is also called as generator potential. It develops when the stimulus is applied, and it is the potential difference developed between the membrane of the receptor. This shows you the schematic diagram of generation of receptor potential and conversion of it into the action potential. Example, pressure stimulus is applied and it is sensed by the Pacinian corpuscle, which is the receptor for pressure. Change in the shape of the Pacinian corpuscle, and there is deformation in the central nerve core fiber, opening of mechanical gated sodium channels, and there is influx of sodium ions, and the receptor potential is produced. There is a local current produced, and the voltage gated sodium channels open. There is further influx of sodium ions. Finally, there is generation of action potential in the nerve fiber. The final property is law of projection. It states that anyway in the path of the sensory pathway, that is from receptor to sensory cortex, anywhere the stimulus is applied is always referred to the receptor location. Example is phantom limb, also known as missing limb. Example, a leg is leg is amputated, the cut end of the nerve fibers are merged in that region along with the scar. Now during the movement of its thigh, would the patient senses as it is from the missing limb. Sometimes he feels pain through that missing limb, called as the phantom limb pain. Now we'll see about synapses. Synapse is a junction between a neuron and a neuron and a muscle, and a neuron and a gland. It is classified into anatomical and physiological classification. Anatomical classification includes between a neuron and a neuron, and a neuron and a muscle, and a neuron and a gland. Between a neuron and a neuron, it is axosomatic, axodendritic, axonic types. Now physiological types, they are electrical, chemical, and conjoint type. In electrical synapse, the transmission is through gap junction. It is bidirectional. And in chemical synapse, the transmission is through neurotransmitters. It is unidirectional. And in conjoint, both electrical synapse and chemical synapse acts. Structure of a chemical synapse: it contains synaptic terminal, presynaptic membrane, synaptic cleft, and postsynaptic membrane. The presynaptic terminal contains the vesicles which release neurotransmitter, and the synaptic cleft contains the neurotransmitter released and the neurotransmitter degrading enzymes. And the postsynaptic membrane contains number of receptors to which the neurotransmitter binds. There are two types of chemical synapse, type 1 and type 2. The following shows you the differences between the type 1 and type 2. Now we'll see about the properties of synapse. First one is Bell-Megendy's law. It states that the impulse is transmitted in only one direction, that is from a neuron to another neuron. Second one, synaptic delay. There is always a delay during synaptic transmission. It is because of release of neurotransmitter and passage into the synaptic cleft and the action of the neurotransmitter. It is normally between 0.3 to 0.5 milliseconds. Now fatigue occurs because due to the depletion of the neurotransmitter, and also due to the continuous stimulation, there is delay in the synthesis of neurotransmitter. Next one is summation. There are two types of summation: spatial and temporal summation, which was told before. It is an additive phenomenon in which there is production of action potential. Convergence and divergence. It means that in convergence, many presynaptic neurons converge into a single postsynaptic neuron, and in divergence, one presynaptic neuron terminates on many postsynaptic neurons. Now we'll see electrical property. There are two electrical properties, excitatory and inhibitory. First we'll see excitatory. In excitatory function, excitatory postsynaptic potential is produced. Now we'll see about how this EPSP is produced. In presynaptic neuron, the arrival of action potential in the axon terminal makes the calcium channels to open, and there's influx of calcium ions into the nerve terminals, and the vesicles now exocytose, and there's release in acetylcholine, passage of acetylcholine through the synaptic cleft, and now the acetylcholine binds to the receptors in the postsynaptic membrane. Ligand gated sodium channels open, and there's influx of sodium ions, and there's development of EPSP produced. Now the voltage gated sodium channels is open, and there's further influx of sodium ions, and there's action potential produced. And finally there's spread of action potential in the postsynaptic neuron. EPSP is like a local potential. It is non-propagated and it does not obey all or none law. Now we'll see inhibitory function. The first one is postsynaptic neuron inhibition. Since it is inhibitory function, inhibitory neurotransmitters are released. First in the presynaptic neuron, when there's action potential received in the axon terminal, opening of calcium channels, in the presynaptic membrane, and there's calcium influx, and the vesicles rupture to release GABA, which is an inhibitory neurotransmitter. It is released into the synaptic cleft. Now the GABA binds to the receptors in the postsynaptic membrane, which makes potassium and chloride channels to open. There's efflux of potassium ions and there's influx of chloride ions in the postsynaptic neuron. Therefore, there's more negativity inside the postsynaptic neuron. This leads to hyperpolarization due to inhibitory postsynaptic potential. Finally, inhibition of synaptic transmission. Next one is presynaptic inhibition or indirect inhibition. In presynaptic inhibition, there's blockage of calcium channels, there's opening of potassium channels, and there's efflux of potassium in the presynaptic neurons, and the neurotransmitter GABA binds to the receptors in the presynaptic neuron itself, and there's inhibition in the presynaptic neuron. Next, Renshaw cell inhibition. It occurs in alpha motor neurons. Here, the neuron inhibits the neuron which excited. This inhibition serves to limit the excitatory function of the motor neurons, and that's why it is also known as negative feedback inhibition. Now, feed-forward inhibition. In this, a presynaptic neuron will excite an inhibitory interneuron, and this inhibitory interneuron will inhibit the following cell. In this way, the synaptic transmission is inhibited. This occurs in the cerebellum by the granule cells and the Purkinje cells. Next is reciprocal inhibition or reciprocal inhibition. That is, when a group of muscle is excited, the antagonistic muscles are inhibited.